Please note today's session is being recorded. If you have any objections, you may disconnect at this time. Thank you very much. And on behalf of the National Cancer Institute, I wish to welcome everyone to the Precision Medicine and Population Health Interest Groups webinar, The Intersection Between Precision Medicine and Implementation Science. A brief word about logistics and we'll be off. We ask that if you are not already on mute, to please keep your phone on mute for the duration of today's presentation. As mentioned, the session is being recorded, and muting all lines will help us to avoid any background noise. We encourage questions. They can be submitted by using the Q&A feature on the right-hand side of your screen. Please type your question in the provided field and hit submit. Feel free to submit your question at any time, but we'll be opening the session for questions when both presentations have finished. Without any further ado, it's my pleasure to turn the meeting over to Dr. Maureen Curry.
Good afternoon, everyone. This is Maureen Curry. I'm the director of the CDC Office of Public Health Genomics and a senior consultant at the National Cancer Institute Division of Cancer Control and Population Sciences. It's my pleasure today to introduce our two speakers, Dr. David Chambers and Jeff Ginsburg. You have their online bios online, but I'll do a brief introduction. As a point of reference, this is part of a regular, probably every other month webinar series. The next one will be on October 19. We will have more information on that on our website. So there's been a lot of conversation lately about precision medicine as the Precision Medicine Initiative was launched last year and how it might intersect with the field of implementation science. And we're really lucky today to have two pioneers, two leaders, one in implementation science and one in precision medicine and genomics. And we're hoping this dialogue will engender discussion. We're giving them 20 minutes each, and the last 20 minutes will be for questions. Feel free to send your questions online. We'll take as many as we can in the last 20 minutes. So without further ado, I'll introduce the two speakers quickly and then turn it off to David Chambers, our first speaker. So David is the Deputy Director for Implementation Science in the Division of Cancer Control and Population Sciences here at NCI. He manages a team that focuses on efforts to build and advance the field of implementation science through both funding, opportunities, training, dissemination platforms, and enhancing partnerships. David has been really one of the driving forces of implementation science across all of NIH and beyond, and he was at the National Institute for Mental Health for many years before he joined NCI. And our second speaker, Dr. Jeff Ginsburg, who's the director of the Center for Applied Genomics and Precision Medicine at Duke University School of Medicine. He's also connected with the School of Engineering. He's professor of medicine and pathology and biomedical engineering. Dr. Ginsburg is a world leader in genomic medicine, interested in the translation and application of genomics in day-to-day healthcare. You can check his long bio online if you want, but he's also well known for his work and leadership in many activities around NIH. He serves in the advisory committee for the director of NIH. He also chairs the National Academy's Roundtable on Genomics and Precision Health at the National Academy of Health. So without further ado, I'm going to turn it to David to kick off the first 20 minutes, and then Jeff. Thank you.
Okay, thanks, Maureen, and thanks, everyone, for tuning in. What I'd like to do with the next 15 or 20 minutes is make the case that implementation science and precision medicine really have opportunities to be mutually beneficial. Whether we think about implementation science historically as the way in which we're trying to improve care, bring evidence, and bring the results of research to those who could benefit from it most, and we think of precision medicine really along the same lines of trying to figure out how do we optimize care and how do we optimize health and healthcare for each individual. And I think the two really can go hand in hand. So what I'd like to do briefly is just give some orientation for folks who are a little bit new to implementation science of what we mean, talk a little bit about a few examples of how implementation science might be considered within precision medicine, and then finish with some assumptions that I think at times hold us back in terms of really trying to optimize the use of evidence, and in this case, ultimately the use of precision medicine findings toward the benefit of health and of health care. So what you see in front of you is an effort by Bayliss and Boren back in 2000 to try and reflect what happens if we assume that the end product for our research really is that publication, and everything magical will happen down the road for making sure that that publication makes a positive benefit on people's lives. So on the left side, what you see walking through are the steps in which we're losing valuable research findings, whether positive or negative, if we're assuming that the publication pathway is the route to implementation. You'll see that results may be negative from research. We may submit papers for publication. They may not be accepted. They may be published in places where they don't join bibliographic databases. They may not be considered in systematic reviews, in guidelines, in textbooks, and they may not make it all the way toward implementation in the community or in clinical settings. On the right side, what you see is just on average their effort to try and map out how long it takes for this process in sort of reasonably best case scenario to get from the conclusion of a research study to implementation. What you're probably more familiar with is this tagline that it takes 17 years to turn 14% of original research to the benefit of patient care. A lot of times people are using this statistic without knowing where it came from. For those of us who are focusing on implementation science, it says to us that we can't rely on our traditional methods of dissemination to assume that findings, that evidence-based practices will be implemented. Too often, we're assuming on the scientific end, this, you can probably in your offices or wherever you're watching this, fill in the rest of the statement. We assume that if you build it, if you create the best possible tools, intervention guidelines, then people will come. But as you can see from this lovely but empty baseball diamond, it's not enough to just bring things and make things presumably accessible to people, but it's about what the demand is for them, and it's about whether anyone actually wants to use and is able to use what you're creating. So for genetic testing, even if we assume that genetic testing in terms of the evidence reaches the point where we can identify the optimal treatment for a particular illness, where we can reduce risk for health problems, when we start to look at that pathway around implementation, we can see how the benefit starts to decline. So let's assume that of insurers who are able to support the payment for care, let's say only half of the insurers are choosing to provide those wonderful tests. Let's assume that only half of the systems are going to go that next step and say, how do we train clinicians so that the genetic tests are potentially, we know when to offer those. Let's assume further that, within those clinicians at those systems who are trained to use these tests, only half of them are prescribing them. And then we assume that the eligible patients who might benefit from this testing, only half of the patients that those clinicians see get tested. Now, it seems like we're making some pretty strong assumptions about the limited use, but in many interventions, if we could get half of insurers, half of systems, half of clinicians, and half of those patients to get those evidence-based interventions, we'd be doing a lot better than we currently are. But if we just assume that sort of threshold, even within that scenario, there's no problems with access. There's no problems in terms of the follow-up, or there's no problems in terms of following the procedures for testing. We're already losing probably 94% of the benefit of that genetic test. In other words, the amount of benefit that we have from getting implementation right may very well outstrip the amount of benefit that we got just by assuming that that test exists. And we need to do better in following that pathway and really understanding implementation so that we don't doom things to sit on the shelf or to be located, tests to be located in a place where they're not used, they're not known when to be applied. So to orient folks, this is how we think about three terms that really underscore a lot of our work in the field. The umbrella term, which we use within our implementation science team here at the NCI, focuses on implementation science as this broad study of how we promote integrating research findings and evidence into healthcare and health policy and practice. We at NIH at times have chosen the words dissemination and implementation research also to map the space. And the reason why we also use these two terms is because we recognize that traditional dissemination methods, which are typically passively providing information through a journal publication, through a message, doesn't necessarily fit nicely to the audiences that might benefit from it. And what we need in dissemination research is a much more robust knowledge base of how we target that distribution of evidence, how we target that distribution of effective interventions to particular audiences so that we're able to spread, but importantly, sustain knowledge and the interventions that could benefit people. We make a contrast between that and implementation research, which we see as much more of an intensive and an active process of trying to bring interventions and other tools to community and clinical settings so that we can improve patient outcomes and ultimately so we can benefit population health. I mention these because there are terms that are frequently used, but often it's not clear how they're being used, and we want to make sure in our efforts here that people know where we're coming from. What do we mean when we say implementation science? What do we mean when we say dissemination and implementation research? So on dissemination research, we're really thinking about that process all around the creation, through the transmission, the reception, and what happens with information as it's translated ideally into positive action toward health and healthcare. There are many different target audiences. We think about our consumers, patients, and families. We think about our providers. We think about administrators, payers, et cetera. And we need to do a better job of understanding the implications of how evidence is created, how it's packaged, and then how it is transmitted to those who might benefit from it. When we think about implementation, we're really trying to draw a contrast between what often we're focusing on, which is what do we do when confronted in a sort of ideal situation with the patient for whom we can try and help a health problem or potentially trying to reduce risk. So often as you look at this cascade of different outcomes, we're really focusing on the left end and the right end. We're focusing on what do we do? What are those empirically supported treatments? What are those efforts to try and improve quality of care for the individual so that on the right end, we're improving function, we're reducing symptoms, we're improving health status. And so in a lot of our trials, we really do see that usual study focusing on what is the intervention and what are the health outcomes that it's going to generate. What we end up missing is all of this important activity that goes in between, where we're not just saying what are the treatments, what are the preventive strategies, what are the other methods and tools that we need to use for the individual, but how do we actually get them into practice? And so the core of implementation is often focusing on what are the implementation strategies that are enabling those evidence-based practices, genetic tests, et cetera, to make their way to the benefit not just of individuals, but of the population. And what we argue is that there are key implementation outcomes like the feasibility within a particular setting of an intervention being delivered, a penetration rate, how far it gets through a particular system, how many people within that system have access to it, sustainability, level of uptake of a particular intervention. And our argument is that if we can improve implementation outcomes, that we'll see knock-on benefits on the service system, and you'll see in this picture the IOMs or classic standards of care, efficiency of a system, the safety, the effectiveness overall, equity, et cetera. And the argument is that if we can get implementation right, we improve the service system, and then we see health outcomes improve, not just one person at a time, but at that population level. So if we're successful with implementation, it's not just that we get these improved implementation outcomes for their own sake, but that we're ultimately seeing large drivers to improvement at the population, at the system level. So dissemination implementation research really does borrow tremendously from a whole range of fields. You'll see some of them listed here in terms of theories, in terms of some of the findings, in terms of the methods that we use. It often requires pretty intensive collaboration with stakeholders from multiple practice settings. I think you'll hear from Jeff some specific examples of ways in which they've developed tools that really do cut across different stakeholders within sort of the genetics and genomics area. It features a whole range of different methodological approaches, and we specify that methods need to be both rigorous but also relevant within the settings where they're being tested. And that studies of dissemination and implementation should really focus on not just do we get successful uptake or improvement in the implementation or sustainability of various interventions, but really trying to understand how and why they work. So we do have current funding announcements for anyone who hasn't been aware of this. At NIH, it's R03s, R01s, and R21s. The National Cancer Institute is at the helm, but many other institutes, you'll see here Fogarty, the National Institute of Mental Health, Heart, Lung, Blood, and our Human Genome Research Institute collaborators, as well as the Office of Behavioral and Social Science Research. It has been our way in terms of advancing the research agenda for dissemination and implementation across the agency. Over 150 grants just funded through these announcements and others from elsewhere, and something that our Center for Scientific Review at NIH has deemed important enough so that there is a standing study section for anyone who is interested in this. So whether you're focusing on these specific announcements or you're identifying your own investigator-initiated research, in this case that cuts across implementation science and precision medicine, there is a logical and an expert home for you for review. So I wanted to just walk through a couple of examples. If we think about Lynch syndrome and the recognition that there are populations within this country and around the world that have elevated risk for a range of different cancers, it's not just enough to know that we have a particular test. We need to be thinking about how do we really enable a population benefit if we identify someone who carries a particular mutation around Lynch syndrome. And so when we think about the implementation science challenges, beyond just the existence of the test, we're really saying how do we take the power that we may have to identify Lynch syndrome and bring it through that broadest possible population, that population of everyone who has colorectal cancer, to bring it to those who are members of the family of those who have Lynch syndrome. And how do we really think about the strategies through which we're implementing that screening, we're thinking about cascading, we're thinking about ongoing screening and monitoring once someone with Lynch syndrome has been identified. So again, really thinking about those other aspects that optimize reach, that optimize the sustainability of a system that's able to capture the population. Similarly for BRCA1 and 2, beyond just recognizing that there are elevated risks, how do we really bring that to the population level? How do we think about some of those scale-up across families decision-making and those procedures? How do we think not just about the testing, but think about the cascade of different services that need to be provided in order to make sure that that test yields benefit to those individuals who may have that particular mutation? And really thinking about what are the implications that it has if we're thinking at that broader level about screening to everyone who might benefit from it, of monitoring, of preemptive treatment if that ends up being the decision-making. And in all of these cases, how do we not just think about the sort of under-treatment, the problem that we're not getting enough access, but potentially issues of over-treatment where you may be recognizing people and providing services that may not be as indicated for them. For the third example, which is just broad precision medicine, tremendous amount of interest and tremendous amount of potential to really try and say how do we do a much better job of customizing treatment, customizing our ideas about health and healthcare to every individual on the basis of a whole range of data that we're now able to gather. But we really need to be thinking, or ideally we would argue be well suited to be thinking about how clinical practice can incorporate these different findings. How do we think in this case about an evidence base that we know that's going to be evolving, and how do we plan for a system that's able to receive that evidence as it evolves, not just assume that either we're waiting for all answers to be in, or that the answer that we get is going to be the final answer on a particular question. How do we think about the workforce that ultimately would be having to implement precision medicine findings? What are the services that are going to be covered and paid for? All of these are the implementation science challenges that go beyond that initial finding, and the more that we have a group of people who are really thinking about anticipating that, we would argue the better off we're going to be. So I'm going to finish with just a couple of the traditional assumptions that we have around evidence-based practices and argue that maybe precision medicine is an opportunity to move beyond them. So often in our traditional way of thinking, we create evidence, we create evidence-based practices, and they're more or less static. They're manualized, they're fixed, and now we have to implement them. Now we have to apply them. Also that our systems, once we understand how our health systems work, they're more or less static. We think we know exactly what needs to be done. That implementation often is this sort of one practice or one test at a time, that consumers and patients, well, we sort of know that there's a lot of heterogeneity, but we're often assuming that if there's an evidence-based practice, it's kind of a one-size-fits-all. And we continue to assume that if someone chooses not to implement various tests or various interventions, they may be irrational because we have the evidence. So the question here is that whether these assumptions relate nicely, if we're thinking about implementing precision medicine where we know there's going to be a lot of change, ongoing discovery, and ongoing need to figure out how do we best apply that discovery. Choosing not to implement, this notion that it's irrational, says that we're not looking at questions about actionability, right? The degree to which a particular finding has a particular action that can be taken to promote or improve someone's health or maintain someone's health. It says that we're not thinking about these challenging issues about reimbursement. It's that we're not necessarily thinking about whether we have the necessary resources around counseling, whether our existing tests are optimal, given that we're seeing a whole range of new tests crop up month by month, year by year, and it assumes that there's a demand for the knowledge, a demand for the evidence that, that there may not be. I would also put up this slide, which says that there might be some rethinking that we have to do in terms of sustainability. Classically, we're thinking about sustainability as trying to keep our interventions and our systems the same for as long as we possibly can. On the left side, and you'll see the link at the bottom, what this is showing are the Beatles. So folks who know the Beatles are seeing the Beatles haircuts, this is the musical group, from 1963 to 1970, and what it underscores is how radically their appearance, and some would argue their sound, changed over time. Yet from 1963 to 1970, most people would agree that this was still the Beatles. Can we think about our interventions, and specifically genetic tests or other precision medicine tools, as going to evolve and allow them to evolve? So should our existing interventions be those that we're assuming should be sustained in the same form, or how do we think about the evolution of these different tools over time? And how do our systems cope with the reality that this field is constantly changing? Where do we go from here? So we've been, in the last couple of years, trying to think conceptually, and we'd love to certainly hear from Jeff and from questions and comments about how do we make this as concrete as possible, conceptually about a different approach to sustainability. In this particular paper, which we did a couple of years ago, it posits that interventions are like puzzle pieces that we should expect are going to change their shape over time, that the context, the setting in which we're trying to use these particular tests is also changing over time. And rather than assuming that we have the best possible benefit from the beginning, are there ways in which we can assume we're improving quality at all points, and ideally quality improved in one setting can contribute to a broader evidence base? For us, this looks a lot like the learning healthcare system, which we've seen the National Academy of Medicine, or IOM, talk about in recent years and a number of people embrace, which basically says, can we not see research and practice as separate entities, but how do we pull research and practice together? This particular paper that we wrote earlier this year was specifically in the behavioral healthcare system context, but the argument was that we now have technologies, we now have data that we're able to gather in a real-time fashion, that if we can figure out the right way to be able to aggregate that, to try to learn from the local lessons of how we apply these very challenging and very personalized tests, might we ultimately have better supports for decision-making? Might we ultimately have better ways to use that data to have accurate risk prediction over population levels? Can we do a better job with care management? Can we do a better job in assessing performance? A paper that Muin and Greg Firo and I had published earlier this year in JAMA was just briefly trying to say, look, we think that implementation science, that learning healthcare, and that precision medicine are three nice complementary things that for too long have sort of been in an isolated manner, right? That there are people who are advocating for a learning healthcare system separate from those who are really thinking about the benefits of studying implementation, and separate from those that are thinking about precision medicine. What we really see is that there can be such this wonderful opportunity to use, for implementation scientists, to use precision medicine as a chance to really be thinking about the dynamism of our evidence base, the dynamism of our evidence-based practices, and in turn that precision medicine can look to implementation science to ask the so what questions. So we come up with this wonderful new insight. How does it actually tangibly make a difference to the population? So the hope for the future is that we align those of you on the call, those of you who are interested in this area, to say how can we most sort of complement what each other are doing. And that, as a final note, that for implementation and precision medicine, we see that context in both cases matters. It's multi-level. That we can't just focus on in a pristine setting whether the practice works, but how well can it be delivered in a whole range of settings. We do have a knowledge base around implementation that continues to grow. It's a team sport, and we need all of you who are involved in this conversation and all those who are not yet to really come together around identifying both the questions and the studies that will get us further along.
Thank you very much, David. Now I'd love to hear from Jeff Ginsburg. Thank you.
Thank you. Can you hear me okay?
Yep, you sound great, Jeff.
Okay, perfect. Can I get my slides up?
Yep, Jeff, if you want to go ahead and navigate, you have control of the meeting, so you're able to move those forward.
Oh, okay. Sorry, I didn't realize it was one deck.
Not a problem.
Okay, perfect. So thank you, Muin and team, for having me here today. I'm wearing 3 hats. One, my role at Duke in Applied Genomics and Precision Medicine, also as a principal investigator in the NHGRI IGNITE network, and thirdly, as a co-chair for the Global Genomic Medicine Collaborative out of the National Academies of Science, Engineering, and Medicine. And I'd like to say that I really don't consider myself to be an implementation scientist, so I'm really privileged to be on the same panel as David Chambers. And what I hope to do in the next 20 minutes or so is highlight some of the efforts that will bring David's theoretical constructs that he so elegantly told us of, to practice. These are my disclosures. I have no conflicts with the current presentation. And I'd start off to say that I think probably most of us that are listening today have been consumed and somewhat obsessed by the challenge of bringing genomic information about an individual to their clinical care. This to me forms the basis of what we now call precision medicine and health. And often we, like in slides like this, depict this as a nice linear relationship going from genome sequence to healthcare. But I would argue, and I think we've already seen evidence of, that it's a decidedly nonlinear path by which we can actually bring the knowledge of the genome into the care of individuals and populations. So this is a diagram that's probably familiar to many of you. It's a framework for translational research that was initially proposed out of the IOM by Crowley and colleagues, illustrating 2 blocks, 2 challenges in translational research, T1 and T2, and then was expanded upon by Muin, Khoury, and colleagues, depicting what was once a highway as a virtuous cycle of translation, beginning with basic discoveries at the bench, moving to human applications, generating evidence in T2, and disseminating and implementing that evidence-based practice in T3, leading to public health and population benefit demonstrated through effectiveness and outcomes research. But to put this into context, I would start perhaps with one of the earliest elements of precision medicine, the use of risk calculators for the evaluation of patients for cardiovascular risk. And it might be worth thinking about how something as perhaps as validated and as simple as this, how has this been actually brought into the practice of medicine, and how is it used? Several papers have evaluated the use of the cardiovascular risk calculators by a variety of physician communities, and the results are not as nice as we would like to see, with a paucity of primary care physicians actually reporting that they actually read clinical guidelines very carefully, and an equally small percentage of individuals who actually use such a highly validated risk calculator in day-to-day practice, owing to variability in knowledge, beliefs, practice patterns among these family physicians. So illustrating some barriers that we might anticipate being relevant to genomic medicine and precision medicine, having to do with the knowledge and literacy, the understanding of the validity of the tools that are being used, and also elements of integrating this into the clinical workflow. otherwise known as being something that will take away from other potentially competitive activities in their practice. Perhaps another example illustrated here from oncology. As many of you know, EGFR was discovered as a biomarker for a non-small cell lung cancer in the 1980s. It was associated with lung cancer prognosis in the 1990s, and it was demonstrated to be a biomarker, a response marker for therapy for non-small cell lung cancer in 2004. And studies have been done to look at how the EGFR biomarker and testing has been implemented into practice. And while data from Boston, Dana-Farber Cancer Institute in particular, show that many of the patients presenting them with non-small cell lung cancers have actionable mutations, in the general community practices or general oncology practices in the U.S., less than 20% of those patients are actually having EGFR testing, and I guess I would argue that at least these two examples may be examples of some failures that we have to truly implement appropriately to bring these innovations to clinical care. Several studies may have even pointed to one of the reasons why this is the case. One issue might be the recognition that until recently that implementation science was a true science and should be supported by national funding agencies. These two studies looked at the distribution of funding for genetic research across both the NCI as well as the NHLBI, and as you can see, the vast majority of our research dollars are going to T0 and T1 research, and a paucity of our research funding is going to research beyond T1. So therein may lie at least some of the explanation for our failures to implement. Another issue may be having to do with payer reimbursement for genomic technologies, and we often think about payers as being sort of a singular entity, but in fact, these data show that there's quite a diversity of strategies and acceptance of evidence across the payer community. In this particular case, the adoption of reimbursement policies for the Oncotype DX cancer prognosis test was highly variable and only became more adopted over essentially a five-year period. So there's clearly variability in the thresholds of evidence needed to implement across an important constituency in our clinical care and healthcare delivery environment. And one might assume that FDA approval of tests as well might be concordant with coverage decisions for tests, and these data indicate that's not necessarily the case, again, across a number of cancer-related genomic tests that are available commercially. One might even conclude that there is almost a discordant relationship between FDA approval and coverage decisions, perhaps indicating that the efforts to achieve one might be somewhat orthogonal or quite different from the other, and I think it's probably fairly clear that FDA approval evidence and payer reimbursement evidence are not necessarily the same, and perhaps seldom are. So to examine the landscape of genomic medicine implementation, in 2012, the NHGRI convened a meeting of United States centers and institutes that are carrying out genomic medicine programs, really to find out what's been going on in this space, most of which was essentially organically developed, and to begin to decide on funding strategies based on these types of data. Just in qualitative terms, what we learned through that first meeting was that there's a lot happening in this space, but it's largely happening in isolation. There was a fair amount of redundancy, and the organizers and the participants of this meeting distilled the findings to the lack of evidence for value for genomic medicine technologies as being a significant barrier, the ability to interpret variants, the lack of knowledge and expertise, absence of standards, electronic medical record integration, and the need for a financial model that might point to sustainability and adoption easier adoption and sustainability. Perhaps one of the most successful programs in genomic medicine coming out of the NHGRI's portfolio has been the Electronic Medical Records and Genomics Network, or so-called eMERGE network, that's illustrated here. While this started off as a discovery program, I think it's rapidly evolved into one that's implementing, particularly owing to its emphasis on clinical decision support. So it has used the EMR for GWAS discovery to do construct electronic phenotyping algorithms, to streamline informed consent, to engage the community, to standardize elements of data privacy, to reach the pediatric population with a flagship program in pharmacogenetics and genomics, educational platforms, and I think centrally to this is the ability to use clinical decision support tools as a primary strategy for implementation. And this is just an example of one of those clinical decision support alerts that comes up in the Vanderbilt Health System when a physician orders Clopidogrel. This alert tells the treating physician or provider what the genetic background of this patient is, and in this case, this is somebody who would be a poor metabolizer of Clopidogrel, would be at risk for thrombosis in the setting of percutaneous intervention. An alternative medication is actually recommended, and in a learning health system type of strategy, if the alert is overridden, the alert actually asks for the reason why, so the feedback can be obtained. And this has been the need for these types of tools to truly implement genomic medicine strategies has been voiced by a number of communities, leading to the development of an action collaborative at the National Academies of Medicine, formerly IOM, focused on the integration of genomic information into the electronic health record, because this has been raised as such a critical issue for implementation. So for the last 2 years, a group of electronic health record vendors working closely with the academic community has really been working on the standards for integration of genomic patient data with other healthcare data in the electronic health records so that it would become routine to deliver that information to providers and patients in the context of patient care. About 6 or 8 months ago, DIGITIZE, which is the acronym for this group, began to publish standards for genetic information integration, the EMR. On the left-hand side of this slide are just indicating the stakeholder groups that developed these standards, and on the right-hand side of the slide is the first implementation guide that was published concerning the use of genetic variants for HLA-B5701 in the context of a Bacavir treatment or TMPT in the setting of mercaptopurine. So I think these types of guidances and the ability for the communities to come together to develop these standards is one of the linchpins for some of the challenges that we see in implementation of genomics into medical practice. The other major National Human Genome Research Institute initiative for implementation of genomic medicine is the establishment of the IGNITE network. It stands for Implementing Genomics in Practice, as you can see in the logo on the lower right-hand side of the slide. This is our first network-wide publication describing the network and the various programs that are in the network. The goals really to link genomic medicine efforts, hearkening back to our meeting in 2011 or 2012, that we were working more in isolation to really develop best practices around implementation, and particularly across diverse geographic and population settings, to begin to contribute to the evidence base for outcomes that would encourage adoption and to disseminate best practices for implementation of genomic medicine and its sustainability. So certainly embracing many of the elements that David talked about as part of IGNITE. This is the network I lead along with Lori Orlando, the Duke Program in Family History. We're also the coordinating center. The Mount Sinai group is working on APOL1 genetic variation in practice to manage hypertension and chronic kidney disease. There are several groups at Florida, Vanderbilt, and also in Indiana that are doing different flavors of pharmacogenomics with germline as well as cancer-related work. And the University of Maryland is working on the use of genome sequencing for diagnosing monogenic diabetes. So this network is clearly focusing on implementation and effectiveness, also on clinical utility, using highly validated genomic medicine tools to achieve this result. This is more or less a template for how many of the studies are being done. Pragmatic trials are being conducted in community-based as well as academic-based practices, comparing genomics informed care with standard of care and measuring a series of outcomes across the areas that I've already mentioned. And this slide just illustrates the Duke project in family history. Just to give you a quick snapshot, we've developed a web-based family history tool that encourages or requires the patient enter the information about their family history prior to seeing their provider. These results are sent to a clinical decision support engine that uses guideline-driven rules to provide a report to the patient and to the physician. So when the patient-provider interaction occurs, it's really about understanding risk and a strategy to implement to actually undertake appropriate screening behaviors. What's particularly, I think we're proud of in this particular project is, and what's been a linchpin for it, is the use of the Smart Fire platform, which stands for Substitutable Medical Applications and Reusable Technologies. So the SMART are essentially apps, and FIRE, which is fast health interoperability resources, is what really allows tools like this to be integrated across multiple electronic medical records. And this particular program was highlighted at the White House last summer at the Champions for Change event because of its use of the Smart on Fire platform and the ability to realize patient-derived information appearing essentially in the electronic health record. So as David mentioned, the IGNITE network is steeped in using implementation science strategies, both pre-implementation assessments of the status of the practices that the genomic medicine tools will be placed into. During implementation, a number of variables are assessed, and ongoing assessment in the post-implementation phase to understand the true effectiveness of that implementation strategy. So these are really, at least to the first of our knowledge, a network-wide utilization of standardized implementation science methodologies, much of which are require the use of qualitative interviews and assessments of the providers, patients, and their environmental surroundings. So I'm not going to go into the details of this, and you've heard some of this from David, but we truly think about outcome measures for implementation science, about how much we've reached the patient population intended, how much the provider and clinics are actually using the strategies and how appropriately they are, and of course measuring both the time and cost to implement. We also are measuring what we're calling effectiveness outcomes at the same time, and what this slide is indicating is that we're really looking at effectiveness at the patient, provider, and system level. We also have a number of different contexts ranging from more standard clinical variables, financial variables, but also psychological or psychosocial and behavioral variables, some of which we can capture directly from the electronic medical record. Those are illustrated in blue. And the others that we are capturing through survey, more qualitative interviews and surveys. So what IGNITE has at least identified initially are several challenges and also has provided some solutions around clinical knowledge. We're developing a vast series of resources around educational materials for the targeted clinician, the clinicians that are using it. As I've mentioned, we've developed in many cases integration with the EMR, electronic health records, both on a more national scale, working with the vendors, but also with local health system solutions that have been devised, engaging the diversity of patient and clinician populations, have most of the groups have developed a genomic medicine advisory board that really is meant to link the stakeholders and engage them at every step. And similar types of strategies have been used to help with a problem that is not unique to genomics, but patient recruitment into these studies. The other thing that IGNITE has done is developed a toolbox that can be found on the IGNITE website. The link is at the bottom of this slide. And while this is not an exact representation of what the toolbox looks like today, it's been undergoing a number of important renovations. You will find knowledge and best practices around testing protocols, about clinical implementation strategies in various environments, about open source clinical decision support tools that have been developed both in IGNITE and the eMERGE network, and a suite of educational tools and knowledge for the various stakeholders in the genomic medicine space. So finally, I just want to close in the next couple of slides just to highlight that the point that genomic medicine implementation is not unique to the U.S., not surprisingly. And as part of the ongoing series of from NHGRI's Genomic Medicine Working Group, we hosted a series of meetings of the international genomic medicine community, the first of which was represented in this publication. But we've now have formed the Genomic Global Medicine Collaborative that is really meant to explore the synergies and opportunities for collaboration and implementation of genomics into medicine on a global scale, and really think about how we not just advance genomic sciences for individual health and population health, but also for global health. And it's important because as we look across the globe, there are now, and this is just a snapshot of several large-scale national sequencing efforts. Of course, the PMI is something that looms large for the United States with potential for 1 million genomes, and I'm sure people are aware of Genomics England, the Regeneron Geisinger sequencing program. The Middle East has programs in both Qatar as well as in Saudi Arabia and other European initiatives, many of which were highlighted in a meeting that the G2MC held late last year in Singapore. But to sum up, I think this is a global challenge and a grand challenge for the implementation of genomic medicine. We're clearly going to see on the horizon through the PMI and many of the initiatives that I just mentioned, we're going to have millions of genomes, obviously contributing an amazing amount to discovery research, but also this needs to be linked on the international stage to precision medicine, population health, and global health. And in the middle of this slide, you can see, I think, what at least the global community is thinking about in terms of the 5 major challenges that need to be addressed, several of which I've mentioned, David has mentioned: evidence generation and the economic model that supports sustainability of genomic medicine, the ability to share data across institutions that are working in this space, both data on implementation but also data on effectiveness and outcomes. The incentives to implement need to be aligned across the stakeholders. As David mentioned, there's a significant emphasis that needs to be placed on developing the appropriate workforce, both as implementation scientists but also genomic medicine outcomes researchers. And probably at the heart of this is our ability to really engage the participants in being part of this research effort and ensuring that they have sufficient trust with their genomic data in particular. With that, I think I'll stop and turn it back to the folks at CDC. Thank you. Muin, I'm not hearing anything.
Thanks, Jeff. Sorry about that. A brief note, thanks to David and Jeff for their wonderful presentations. We will now open the session for questions. As a reminder, questions can be submitted using the Q&A feature on the right-hand side of your screen. Please type your question in the provided field and hit submit. Slides to this presentation and the archive will be found on our website, the direct link to which is in the chat box on the right-hand side of your screen. Or not, you can find our website at cancercontrol.cancer.gov. The Precision Medicine and Population Health Initiative is found under cross-cutting areas on our homepage. And with that, I'm going to turn it over to Muin.
And thank you, David, and thank you, Jeff, for wonderful presentations. As we get some of the questions come in, I just want to initiate some discussion here. And, you know, I come from a world of public health that has a healthy skepticism about genomics and precision medicine. And the word that I keep hearing in my circles is that, is it too soon to think about implementation given the status of precision medicine? So that's sort of part A to this. And the part B related to this question, I'd like to ask both Jeff and David, is the Precision Medicine Initiative per se. I mean, you both alluded to it. There will be 1 million or more people enrolled in a cohort. This is primarily a discovery platform. But from your perspectives, what kind of opportunities for integrating implementation science directly into that platform of discovery based on the learning health system model that you talked about? So 2 parts to this. Are we too early to think about this implementation stuff when we're still discovering new things, and how do we meld the 2 worlds in this case of the precision medicine large cohort? Whoever wants to jump in, you're welcome.
Sure. So I can see Muin, so maybe it's easier for me to jump in. So the question about is it too early to jump in around or to be thinking about implementation where precision medicine is concerned? I think if we don't think about the ultimate benefit of what I think is a potentially incredible discovery platform early enough, we're going to replicate where we failed in many, many other cases, assuming that the answer that we create in a vacuum is going to be that answer that fits best for people's lives, for health systems, et cetera. So I don't think it's too early, and I think that just the reality that in various ways and under various names, I would say for a long time, there have been all sorts of efforts to toward trying to improve the precision of medicine, to try to, whether it's specifically with genetics or other ways of trying to understand risk and understand optimal treatment, we should be learning from each and every one of those opportunities. I think often there is that gulf between research and practice that doesn't seem to serve much good, I think, at least over time, if what we want to see is the optimal benefit out of our research. And so the idea of being able to look at ways, whether it's just within genetics, of a whole range of different systems that have been thinking about how do we set up a process through which we can optimize the delivery, the use of particular tests, to optimize the process through which we use that information to drive decision-making. We should be learning from those natural experiments, really, and trying to study them. And to your point in terms of what to think about, I think we know from implementation science, and I'm sure, you know, from Jeff and others' specific experience with eMERGE and with IGNITE, how valuable it is to understand context. And so I saw on one of Jeff's slides where they talked about the variables that they're already measuring to really understand things at the provider level and the system level. And I think within precision medicine, with that initiative, we have an opportunity to really understand more about the context, not just where healthcare is being delivered, but where people are living their lives. If our goal is to try and figure out not just what to do, but how best and where best to intervene, we're likely going to have an enormous amount of data that understands where people are and ideally how best to reach them. And those, I think, are questions or components that could be nicely added to a whole range of different projects.
Thank you, Jeff.
Yeah. Can I jump in on this a little bit too?
Sure.
So first, to your too soon question, I want to echo a little bit of what David just said and maybe maybe say it slightly differently. So certainly precision medicine is not new. Many have argued that that's the way that medicine has been practiced. The tools have been evolving to allow that to be more enhanced. And of course, I showed one example of a tool that's been around for over 50 years that has not been adequately implemented, perhaps. your own work, Muin and others, has identified Tier 1 evidence for certain genomic technologies that should be implemented, but maybe largely are not. So I guess I would say we should not let perfection be the enemy of the good. We should at least use the most advanced tools and evidence where evidence exists and really define that path for implementation that will allow the ones behind it to move forward. I think I'm not sure exactly what we would be waiting for to pull that trigger. Maybe, Muin, you have something in mind, but I think the more we begin to advance the earlier stage technologies that have the evidence base, the shorter the time will be between now and when they have true public health impact. Regarding the precision medicine initiative, I view the PMI, at least as it's being constructed right now, as you said, as a massive engine for discovery. But that engine for discovery is going to lead to a whole series of novel insights and for disease and tools and technologies as well that should be implemented. So again, defining that pathway with the tools that we have now in advance of the PMI will only make the PMI impact, in my mind, more realizable. Whether it's through pilot demonstration projects of the type that IGNITE and eMERGE and other are doing, and others are doing even independently of federally funded programs. But we have to create that framework and create the ecosystem that will allow the PMI discovery engine to coexist with the implementation science engine, and what David and you have published on this learning health system piece is a significant part of the equation. So we can't wait for all these pieces to become as mature as we might like them. We should begin to assemble them now so that the PMI can have its greatest impact.
Great, thank you. So we're going to start taking some of our online questions. The first reads, typically, evidence-based interventions are based off of RCTs. This is not possible in genomic medicine. How can we balance different types of evidence, i.e. observational studies, with evidence-based recommendations?
So I agree with the statement that with the deluge of genomic findings that are coming into play, and actually the pragmatic issue of the cost and feasibility of an RCT, that we can't expect to see RCT level of evidence generation for everything. So we have to achieve that balance. In my mind, perhaps the balance is really around what the risk is around the tool or technology that's in question. So, for example, do we need to have should we have an RCT for a genomic variant that maybe informs a lifestyle choice versus a genomic variant that informs a chemotherapeutic agent selection, where that might be have a significant impact on one's livelihood and future life or death in many cases. So should we begin to align the evidence generation structure with the level of risk of the technology that's being considered?
Yeah, I think that's a great point, and I'll also say that I think to some degree one has to ask the question of why is one choosing to focus on randomization? I think it ends up, I've heard a lot that in order to have sort of precision or personalized medicine, your trials have to be, in order to be RCTs, have to be ridiculously large. It's kind of that counterintuitive thing. If we're focusing on the individual, we get bigger and bigger and bigger. But I guess it ends up being, to some degree, as we're gathering all of this information that really gets to pretty heterogeneous individuals, to what degree does it serve us to try and figure out the appropriate grouping for randomization, because we may have a lot of challenges there. And so I guess often the question ends up being, and these are for our methodologists to help us with, is how can we optimally answer the questions that are provided, and whether that's through a randomized controlled trial in certain ways, or through studying natural experiments, through capturing dynamic information. I think we need to use a full range of approaches.
And just as a brief note, for those who must disconnect at 3 p.m., thank you so much for attending. If you are able to stick with us, we are going to answer the 3 last questions that came into the meeting online. So I'm going to go ahead and read those out. The first reads, should those of us involved in classic technology assessment in medicine think about the use and dissemination of genomic technologies any differently than we have with non-genomic technologies/interventions? If so, why?
I would say I agree with the what's underlying this is that that we shouldn't necessarily consider genomic medicine as an exclusive technology. As a matter of fact, I tend to think that we have not done the same implementation, thought about the same rigor of implementation science with other technologies such as imaging. But the only caveat here is I think is the issue around data security and privacy with respect to one's genomic information, that is genome sequencing information, has another layer on it that is going to have to be addressed to achieve full implementation.
And I think ideally we're using the sort of mutual advantages of implementation science conducted outside of genomics and genetics to improve our understanding there and vice versa. And so I really see it as, yes, there with particular treatments, particular interventions, particular types of data, there are some specifics that one has to be thinking about. But overall, we should be learning some of the same barriers that Jeff had mentioned, as well as some of the same strategies to overcome them are consistent whether one is specifically focusing on genetics or genomics or thinking about other interventions.
Great. Could you discuss a bit more about your thinking about implementation science in precision medicine versus in prevention and population approaches to cancer control?
So I think it's a great question. I think just that overall, I think they're ultimately going to converge, because I think that we've seen, and even from the series that this specific webinar is a part of, just as well as a broader thinking of what precision medicine is, shouldn't preclude the idea that you're focusing at the population level or you're focusing on the prevention side. So I think the key is trying to get a sense of what are the key questions in each area and how do we go about optimally answering them. I don't necessarily think that there is a different world between implementation science in say, genetics and genomics from that in other areas. And I think the benefit of seeing the work and seeing the number of institutes, for example, that are involved in our Trans-NIH program announcement, involved in our annual meetings, is that you see common threads, common discussion across the board. So I don't know specifically from my perspective that there's a totally separate agenda, but rather to say, can in each place, can we be coming together on what are the priorities, what are the capacities that we need to build, and what are the questions that we need to answer?
And I would just say I've learned a lot from both Muin and David about this point. The agenda for implementation science into a healthcare system is one agenda, but the agenda for implementation science into public health to do population, to affect populations is a somewhat separate agenda. They have certain commonalities, whether they have to be done in sequence or can be done in parallel. I tend to believe the latter. That we should be thinking about how to do public health implementation of genomic medicine technologies while we're working with complex health delivery systems. And hopefully, as David said, the two will converge.
This is Maureen here. Just to add to what Jeff and David said, this whole webinar series is part of a long-term dialogue between medicine and public health focused on how to bring precision tools to the population level. And so there is really no different agenda. Sometimes concepts converge, but what we're trying to do is, at the end of the day, better cancer control and prevention, or any other disease control and prevention. And that would be a good segue for our next webinar on October 19, where we explore more fully the idea of precision public health. So stay tuned. So there is one more question before we go.
That was an excellent plug, Muin. And with the last question of today's session, this is regarding the Global Genomic Medicine Collaborative. Excuse me if I messed that up. The question reads, is the Million Genome Project interoperable with the 100,000 Genomes Project? Are collaboration members collecting the same family history data? And it continues to say that's important in rare diseases where our relatives overseas may hold genomic keys.
Yeah, thanks for that question. So I think the key word in this question is interoperable. If I made one believe that there is a million genomes project, that's not really what I was meaning to say. I was meaning to say that all of these independent efforts are going to result in millions, if not of genomes that should be interoperable in terms of the types of data that can be shared across these nations. And the question about whether, all these groups are using the same clinical assessment tools, phenotyping tools, family history tools is a huge one. If we're going to create the global knowledge base for genome-based discovery or implementation, we've got to get the global communities to come together and really agree on the standards for acquiring the data, standards for disseminating data, and of course the tools that should be used for clinical phenotyping. So we're a long ways off from that, but I wanted to at least indicate that we had our toe in the water and that there was a lot of interest and motivation on the part of the stakeholders across the globe to do this.
Wonderful. Thank you so much, and thank you to everyone for sticking with us. I again want to thank Muin, David, and Jeff for their time today, and to you for joining us. You may disconnect at this time.
Thank you.